Let's go to, okay, so let's go to Shoni Jain. Are you here, Shoni?
Yeah, I'm here.
Okay, go ahead, honey.
Okay, I'll share.
Okay.
Okay, I'll present. Okay, can you see it properly?
Yes, it's perfect. Go ahead.
Okay, perfect. So this is my thesis defense. My name is Shoni Jain. So my research topic today is alpha-synuclein targeting monoclonal antibody therapy treatment for Parkinson's disease. And currently I'm a junior at Westview High School in Portland, Oregon. So I researched this topic because I have a keen interest in public health, and these therapies are something that are going to enhance community health, which I really appreciate. I also currently work at a retirement home, so I kind of see people who are affected by this disease on a daily basis. So, the topic area that I'm investigating today is possible immunotherapies for treatment and betterment of the neurodegenerative disease called Parkinson's. And my main research questions that I'm trying to answer is what causes Parkinson's disease? So I'll kind of explain that with our introduction. And then what types of therapies and antibodies can be used to battle alpha-synuclein, which is a big proponent of why Parkinson's occurs. And why is this important? So Parkinson's affects like about 10 million people in our population, and it is a pretty prevalent neurodegenerative disease, especially in elder populations. So if we work towards innovating new therapies like these antibody therapies, we will overall see a betterment of health in society, which I really appreciate. And my main objective is to conduct a study which is investigating a-synuclein and the causes of Parkinson's disease and examining the uses of these antibodies in passive and active scenarios as therapies for betterment of Parkinson's disease in these patients. So first, I'm going to start off with a little bit of background information. Just starting off with what Parkinson's disease is. So it's a neurodegenerative disorder affecting mobility in patients during usually old age. Parkinson's causes a loss of like nerve cells within the brain in an area called the substantia nigra, which is kind of noted here in this image. And this part of your brain is what's transmitting dopamine into the rest of your body, which the lack of dopamine is why kind of like these mobility issues are prevalent in Parkinson's. So if you see in the healthy patient, you can see that the nerve cells are kind of there in the substantia nigra, and while in this Parkinson's patient, they aren't seen. So another part of this, going more deep into it, is there's a thing called alpha-synuclein. So these are within Lewy bodies, and Lewy bodies are abnormal clumps which have this protein called alpha-synuclein, which has been abnormally folded and aggregated, causing like a halt to some neurotransmitters and chemicals. And you can kind of see an image of the Lewy bodies right here and how they occur within our brain and our nerve cells. So this is a lot of information, but I'll summarize it for you. So there's two types of therapies that occur, passive and active therapies. In passive therapies, you have like a antibody or an immune cell factor that you're directly inserting into a patient. And the downfall with the passive immunotherapies is that you are not seeing long-term results because it's not creating memory in a person's immune system. In contrast, though, active therapies are doing this. And what they can be seen in a form of like vaccinations. In any vaccine, like even in like, a COVID-19 vaccine, you're injecting these things in like trying to create have your body to create memory to create antibodies towards an antigen that's occurring. So that's the difference between these two different therapies. So here is my literary review. So a lot of my trials for my passive immunotherapies came from the New England Journal of Medicine, and there was two of them. One was prasinezumab. I'm probably like butchering that, but there's also cinpanemab So these were two of the antibodies that were being studied for passive therapies, and they were targeting different regions of alpha-synuclein. So one was the C terminal and one was the N terminal. And then secondly, I kind of used clinicaltrials.gov to find some few prevalent active therapies as well. So one that I wanted to focus on was PD-01A, also known as Afitope, which was done by the biotech company Gabber. And they made a vaccine, kind of how we discussed with. So talking about my methodology, I came up with kind of researching antibody therapies in Parkinson's after I initially was looking at like the success in Alzheimer's disease, because Alzheimer's has much more of a success rate with like passive therapies than Parkinson's currently does. So I wanted to look more into this disease specifically. And for my research, I used a lot of clinicaltrials.gov and libraries and journals, and went in and found data and statements that supported my question. And then this was all done with the purpose of collecting from previous and current trials, and analyzing what went wrong and what could be done for future audiences to understand. So I'm going to be focusing on the results of the two different types of things. So I was talking about the passive and the active treatments. So for the passive, there wasn't a lot of success that was going on there. We didn't see any kind of effect in the pathogenesis or the progression in Parkinson's disease, which is one of the main factors that we were looking at when seeing, okay, is this affecting? Um, and though we did see some reduction of like motor symptoms that were occurring, the passive treatments, specifically Pasadena, didn't really do much. For the active treatments, we did find that the antibodies grew within the person and that they were affecting what was going on. But even though that happened, they did fall again after 2 years, and the vaccines were had to be re-administered. So what we kind of saw, like as a general summary from that, is that looks like more active therapies are more viable options. Like besides this, I also did more look into active therapies in my paper in more detail, and it seems like active therapies were more predominantly used, and were kind of reducing pathogenesis and possible symptoms, while passive therapies are still kind of a real work in progress, and we haven't seen many definitive answers to that so far. The thing with passive therapies is that they work on mice models, but then there's some translation errors when going to humans, which is why it's not working at the moment. So I think the main way we can see if this is going to work or not is seeing the difference between mice models and humans and where that's going. So my question was like, what types of antibodies and other proponents can be used to battle like ac-nuclein, which is a PD causer. And so it was found that active therapies are more effective than to show a difference in progression than passive therapies are, since they're not showing the desired effects. And so my contribution of my research is that I'm kind of creating like one paper that will kind of exemplify most of the well-known trials and therapies for people who are researching in the future to kind of use to innovate more research and create better therapies for people with Parkinson's. My limitations were that this was online research and I'm a very hands-on person. So I wish I was able to do that, but still I got very substantial information from the literature reviews that I was able to read. And also sometimes the comparison of these therapies was a little bit hard because some were less in depth than others, since this was a very like topic that hadn't been trialed much before. So one of the quotes that really stuck out to me when I was doing my research that I read on the paper was, though this is a dead end, there's always an American land waiting to be discovered by Columbus. And this kind of is telling us that science is always building, and most of these trials still have work to do, and we have to keep that in mind. So maybe in the future, there will be a more definitive therapy for PD. So I want to talk a little bit about my research experience. First, I want to thank Dr. Varkey a lot. So he helped me a lot getting the correct science behind my studies and making sure that I wasn't confusing things. So in our one-on-one sessions, I got a lot of help with that. And he really helped me finding places for my resources like clinicaltrials.gov. I did not know that existed beforehand, and I was super excited being able to like go and read actual trials that were going or had gone on. I also want to thank Professor Virgo for what all he did. He helped us format our paper and make sure we were ethically doing our paper, sourcing the correct people and things like that. And I was able to encapsulate everything because of him. So overall, this neuroscience research program really allowed me to formulate my review paper and understand the research process and how to formulate this paper, because I really had only done that in like biology class at school, and that wasn't as in depth as this was. And I'm so excited that I'm able to like contribute to further innovation that may happen in the future, even if it's pretty slight. So I want to thank you all for listening today. Thank you.
Amazing. You want to turn off your sharing, honey? Yeah.
Yeah.
I mean, this is maturity, right? Quoting Columbus and accepting the journey of uncertainty. A big round of applause to you, Shoni, and the
Thank
work
you.
that you've put in. Love the energy, love the effort that you've put in. Amazing. Keep doing that. Dr. Jobin, please, any questions for
Yeah,
Shoni?
excellent presentation, Shawnee. I see that you put a lot of efforts after that 101, like, and went deep into those, like even the uh, pharma companies who are trying to produce or get these therapies into the market. One of the questions that I have is like when you compare the active and the passive immunization, was it comparison for the same antibody or were those two different antibodies?
Yeah, so I wasn't looking exactly at the same antibodies. I was just looking
Okay.
at kind of in general, like between like passive and active, like which treatment method was best. So it wasn't like specifically the antibodies. Yeah.
Okay. Okay. So that could be, so sometimes in passive and active, that could be a difference in titer, as well as like if you have two different antibodies, it could be the epitope difference itself with respect to the page. Okay. Great
Okay,
job. Good job. Yeah.
thank you. I'll keep that in mind for my paper. Thank you.
So amazing, amazing.